Well, thank you everyone for joining in. Good morning. I hope everyone's doing well. My name is Ezequiel Zylberberg. I'm VP of Product Development and Planning at Akron. And I'll give you a quick overview of who we are as an organization. But what I really want to spend my time on today is talking a bit about a question that's been on a lot of our minds, and it's around supply chain integrity, especially in light of the growth that we're experiencing as an industry, and also as a result of many of the challenges we're facing due to the COVID-19 pandemic. And then really bring it home and talk about how we can work together as an industry to build a more robust supply chain using really the case study of the very core of who Akron is, which is the production of recombinant proteins and cytokines for further manufacturing use. So I'll start with just a very quick overview of who we are as an organization and really who we started as. So we started very much in the realm of cytokine and recombinant protein production, targeting the cell therapy market. And over time, what we've really grown into is an end-to-end service provider, a solutions provider that offers compliant, large-scale solutions to enable commercialization of these advanced therapies. And so we understand a lot of the challenges that cell therapy developers are facing as they advance through the clinical pipeline and require really a more and more robust supply chain and a more integral system for delivering their products. And so we've really built our system from the ground up in order to be able to support that commercial development. So just a very kind of quick overview of who we've been and how we evolved to where we are today. We really started, I would say, on a hunch. Akron was founded on this idea that eventually the quality requirements of the ancillary materials that go into the production of these advanced therapies would eventually align more closely with the requirements placed on biopharmaceuticals and pharma products than on research grade materials. And so from the very beginning, we built our production capacity and our supply chain in a way that would allow us to eventually support the industry once it reached the commercial phase. And that view around building quality and compliance up from the ground kind of carried through as we built out our product portfolio. So we ended up launching a cryopreservation line called Cryosolutions, and this product is now embedded in several BLAs, especially in the US and Canada. We launched a whole family of virus inactivated human plasma-derived products in order to be able to support the safe and consistent production of cell therapy products, and eventually started moving a lot of our cytokines into liquid format, understanding that folks would eventually need to close their manufacturing platforms and be able to support a more integral process. Now, those kind of innovations and those ideas around supply chain resilience that we built in from the very beginning have really only accelerated since our client acquired a majority stake in Akron in 2019. And we've really continued to grow with that same orienting idea that we need to be able to support highly compliant, large-scale solutions if this industry is really going to be successful in addressing medical problems at scale. So we continued by filing eCTD format drug master files, and I'll talk a little bit more about that during this presentation, really to support our immunotherapy core cytokine portfolio, our IL-2, IL-7, 15, and 21. We announced the production of plasmid DNA under GMP compliance. We have a new facility that is coming online this year where we're going to be able to support gene therapy manufacturers and gene modified cell therapy manufacturers with highly compliant plasmid DNA for their production processes. And most recently announced that we have a new partnership with Octapharma that really focuses on the provision of virus inactivated AB serum to support cell therapy production. So very excited about that moving forward, and think it will provide a really new twist on a product that we all really need and use in our systems, but really require a greater degree of compliance behind. So a little bit about supply chain resilience and how we're viewing it at Akron. So I think we have to think about two axes really here. One is resilience is all about adjusting to meet the needs presented by upside events. So right now we understand that the supply chain for cell therapy is highly compressed and requires a lot of investment and a lot of growth, because what we're seeing is an expansion in the number of clinical trials, and what we foresee starting in 2021 and moving forward is the increasing rate of approval of several of these advanced therapies. So we're really going to have to grow to meet the upside associated with that. But there's also the downside, right? We really need to build a resilient supply chain to be able to smooth out a lot of the disruptions associated with things like COVID-19 that have really changed the way that we think about our supply chains. Whether it's Akron, whether you're a cell therapy manufacturer, whether you're a bag manufacturer, you really have to think about this a lot more critically than we used to. Now, upside events. We all know that this industry is growing, and actually, when surveyed recently, more than 50% of biomanufacturers expect really moderate to severe capacity constraints in reaching commercial scale by 2025. We all know that there's a crunch coming. And we're really building up, and you're seeing a flurry of activity these days to build up that commercial ready capacity. But there still is that worry that we won't necessarily be there if we don't start investing now or yesterday. And we also understand that this is going to increase the amount of pressure on the supply base. There's an incredible number of clinical trials, whether you look at CAR-T, NK, the iPSC market. There's a lot of activity happening in the clinic, and more of that is going to edge towards commercial. And so it's going to place a tremendous amount of pressure on the supply base to make sure that we can provide highly compliant, scalable solutions. Now, on the downside, we all know about this. We're all experiencing this. Half of cell therapy companies and a third of gene therapy companies are experiencing supply chain constraints associated with COVID-19. Now, I would argue that the other half and the other 2/3 are experiencing the same constraints, but are not necessarily reporting it. We're all living in the same world where the priority has been given, and in many ways rightfully so, to the production of vaccines and to addressing this global pandemic that we all find ourselves in. But it really creates significant constraints that affect the way that all of us do business. And so we need to be able to adjust quickly to be able to address that. And really a microcosm of that is really the shift in passenger airlines and in traffic and cargo capacity associated with that, although that is coming back up. So how do we think about what it takes to build a resilient supply chain? And I would argue that there are three main items that we need to have in mind. One is standardization. Two is the way that we design our supply chains from the very beginning. And three is regulatory compliance. And I'll talk a bit about each of them and focus in on how, in small case studies and small little vignettes, from a cytokine manufacturing perspective, we're addressing each of these themes. So for standardization, it's counterintuitive in a way, but you gain flexibility by creating a more standardized product because you're able to scale it, you're able to create a more robust system that is impervious to external shocks. So by codifying the rules and the procedures of your process, you're able to permit actually greater flexibility. And what we see more and more is the need to shift from a bespoke supply chain and bespoke manufacturing process and a bespoke industry in many ways, which is what this has grown out of, into an off-the-shelf, scalable series of solutions that are going to be able to drive down costs and make these therapies accessible in large scale. So what we've done at Akron really broadly to support these larger efforts has been very involved on the standardization front. We understand that in order for us to all be successful, we really need to codify the interfaces between us. We need to be able to talk as suppliers of critical ancillary materials to the suppliers of cells for off-the-shelf products, to you, the cell therapy manufacturers, and we need to make sure that we're all speaking the same language. Otherwise, we risk issues in translation, which ultimately are going to impact our efficiency and lead to less than desirable outcomes, so to speak. So just a small case of how we think about this on the cytokine production side. And I'll focus in on this case because I think it's highly relevant, highly important, and relatively poorly understood. But really, there's a great deal of variance in the way that we each assess the activity of our recombinant proteins. Manufacturers will use a variety of methods. And I think what happens at the end of the day is that you are less able to kind of move from one manufacturer to another and achieve consistent results. And that's a problem because, again, if we're thinking about the integrity and resilience of our supply chains, it's very difficult to onboard secondary suppliers and to be able to toggle between different sources of these critical materials if you really have to adjust your entire process once you onboard them. So for us, it's critical that we ensure the consistency and reproducibility of the way that we assay these products, because this will ultimately have an impact on the integrity of the cell therapies that come out downstream from us. So what do we do and how does this differ? So what we know is fairly common in the cell therapy industry on the production of cytokines is that biological activity is typically measured by an ED50 comparison. You basically take the entire concentration response graph, which you can see on the right, and you really measure and connect directly to the specific activity of the protein. But it creates a little bit of variability from one measurement to the next because you're not first assessing the statistical validity of the samples and of your process. On the other side is really the parallel line assay approach, which is what we employ at Akron, which is what the WHO has used, the World Health Organization has used when talking about the standardization of biological activity assays for IL-2 in particular. But we really start with a parallel region of the graph. We ensure that we are passing tests for statistical validity before we actually measure the relative activity of our sample to the standard, and then generate values. And what that allows us to do is generate consistent results from lot to lot. So as a cell therapy manufacturer, you're typically building your process off of a reported activity value on a certificate of analysis that's provided by your cytokine provider. Now, at the end of the day, if there's variability in the way that that assay is run and the results that are reported, regardless of how consistent the product might be behind it, you're going to have to adjust your dilutions and your manufacturing process downstream, and you're going to have a much more difficult time comparing across suppliers. So as I mentioned, the way we approach this challenge is by going to the basics, understanding what is accepted by leading authorities and by standardization bodies in the industry. So we start with our parallel line approach because we understand that it generates a statistically valid result, and that any variability that we'll see beyond that is likely owed to biological variability. The fact that we're working with biologics and there is going to be inherent variability from one lot to another, but we're able to tighten that margin of error and ensure that what we're generating at the end of the day is a sample with, is a product at the end of the day with less variability from lot to lot. So we align our principles of design behind these assays and our principles of analysis beyond these assays in line with the relevant USP chapters around the design of biological assays and around the analysis of these biological assays. So we think that this is incredibly important for generating a robust and consistent product. And ultimately, we think that this is critical for us to standardize among cytokine providers, because this is going to determine how the entire industry is able to grow the availability of suppliers without having to do costly rework to ensure that we're able to work across different suppliers in our supply base. So we think that the standardization is critical. And what you see at the end of the day, and we'll just show briefly, is that our biological activity remains fairly constant across lots. So there's a kind of a selection of a couple of lots here. And what you find is that we're typically around the value of the standard, and that we see relatively little variability. Now, this is owed both to the process that we employ to manufacture our cytokines, in this case our IL-2, but also due to our measurement methods, which I think are incredibly robust and allow for consistency across lots. So I think that this is an important point to drive home. Second really is on supply chain design. This is something that we think about quite a bit, and we think that the industry should be considering more. Now, all of us, when we are assessing a critical ancillary material, really have to make a make or buy decision, what we call in economics. Where does your supply chain begin? And we think that this is an incredibly important competence to have, because ultimately what we see is, sometimes it makes sense to internalize certain activities. Sometimes it makes sense to build incredibly robust partnerships with suppliers. And we think that these long-term, robust, transparent relationships with critical suppliers in the industry is going to be important to really drive downstream success for all of us. And what we find at the end of the day is that very few early stage cell therapy or advanced therapy developers implement a risk-based approach to assessing, onboarding, and maintaining critical material suppliers. And this is something that we all need to work on collectively, because building a robust supply chain and building a robust supply base is going to create redundancy. It's going to create the ability to operate across different suppliers, and it's ultimately going to create a more resilient and secure supply chain for you and for all of us really as an industry. So we think that this is an incredibly important core competency. So how do we address this particular issue? Akron, since the very beginning, as I mentioned, really focused on the production of highly compliant, pharmaceutically compliant cytokines to support cell therapy manufacturing. We understood from the very beginning that the quality requirements placed on these products was going to increase, not decrease over time. And so rather than kind of upgrading our quality system over time from the provision of research grade materials to the provision of GMP materials, we started from the very beginning by operating out of a large-scale facility that has the capacity to do very large-scale manufacturing under GMP compliance. It's audited by national competent authorities, exists under the Pharmaceutical Compliance Scheme, PIC scheme. And really what we're able to achieve there is production under a QMS that can handle the increasingly robust requirements placed by regulators on the supply chain for this industry. Our purification strategies are robust as well, and I think that speaks to the quality of our production system and our production capacity in place. We really employ a tag-free purification strategy, use several different orthogonal steps, and make sure that we have the in-house sterile filtration, lyophilization, and fill and finish capacity to do everything from end to end in-house to provide a highly compliant quality material. So that's really how we are addressing constraints in the supply chain, by producing at scale and in a compliant setting that is going to be able to stand up to the scrutiny that we're all going to face as this industry moves closer and closer to large-scale commercialization. And really, when you look at the specifications that we aim to achieve, especially for our IL-2 product, but also for our IL-7, our IL-15, and our IL-21, we really align ourselves with kind of the pharmaceutically compliant biosimilars that we work towards. So in this case, Proleukin. We understand that it's a pharmaceutically licensed product and it's in widespread use for further manufacturing as well, but we also understand that it's being repurposed for this. And so we're building a IL-2 that is aldesleukin. It's the same product and built to similar specifications, with the idea that we can move away from something that was not suited for this purpose to something that was really built out for commercial production. And that goes through to when you look at the functionality data. So we've done an extensive amount of work with our colleagues over at the Dana-Farber Cancer Institute to really demonstrate that at the end of the day, when you look at the function associated with exposing these different interleukins, so Proleukin and in our case, Acron IL-2, to certain T-cell subsets, we're able to achieve similar degrees of functionality. What that allows us to do is, again, ensure that we have a robust data package to be able to support interoperability between providers, which at the end of the day, again, and it's the theme of the talk, makes it so that we have a more resilient supply chain across different suppliers. And it goes beyond the product itself. I think that a lot gets lost when we think about how the product is presented. So we do present, much like several cytokine providers, our cytokines in lyophilized format, and we adjust really the concentrations and the protein mass to be able to support different processes. But we also have moved quite aggressively towards the provision of cytokines in shelf-stable liquid form. We understand that everyone is moving, and rightfully so, towards increasingly closed processes that allow for the safer production of cell therapies and the more consistent production of cell therapies, which is why we were the first to market with a liquid IL-2 in a syringe, and which is why we're really pushing the envelope with the introduction of single-use bags with shelf-stable liquid IL-2, 7, 15, and 21 this year. So please reach out if you'd like to learn more. This is really an initiative that we're driving in the early part of this year, and look forward to having GMP products off the shelf later this year. So this is going to be, we think, very important for the industry as we move forward into more large-scale production. And lastly, I want to talk about regulatory compliance, because it's increasingly important, and I've mentioned it throughout the talk, but we do need to make sure that we're balancing business needs with regulatory compliance. At the end of the day, it's what we focus on most from a supply chain perspective, ensuring that we are able to make business decisions that not only comply with the increasing needs on the regulatory side, but that also makes sense to provide safe and effective therapies to patients at a cost that is really scalable and makes sense both to payers and to patients themselves. And the way that we've really focused on this is by putting a lot of our CMC information into the Module 3 portion of our drug master file. So we have, as I mentioned, put together ECTD format drug master files for our interleukin-2, 7, 15, and 21. And actually, these files have now been opened by FDA in support of commercial advanced therapies. So we really believe in the integrity of the processes governing the production of our cytokines, and regulators have supported the use of these cytokines in commercial processes. So these files are available for cross-reference by cell therapy manufacturers, and we do encourage folks that we work with to request letters of authorization to cross-reference, because this creates an incredibly robust package once you go to regulators and are able to leverage these filings. And at the end of the day, really, this is the work that goes into building a robust product. We think about the manufacturing process. We think about the analytical package governing the measurement of these cytokines. And we think about the support documentation that ultimately has to go into this file. And what you get at the end of the day is and this is a kind of high-level look at what a drug master file in eCTD format looks like, contrary to the paper-based master files, which are not as let's say expansive in their scope, is quite a robust package that regulators can then look through, assess, and evaluate on the basis of your broader filings to make sure that the items, the critical materials that are going into the production of your cell therapy are of a sufficiently high quality and robustness to really support late-stage clinical development and commercialization. And lastly, I think it's just important to note that we at Akron operate under a QMS that is governed by all of the relevant GMPs for this space. We believe quite a bit, not only in contributing to building these standards and to upholding them, but also to working under them and making sure that we are operating in compliance with what is most relevant and critical for this industry from a quality standpoint. So in summary, I just wanted to bring this back to resilience around the supply chain, which is really what we started the talk with. We really have to make sure that as an industry, we're building a supply chain that is able to adjust to the upsides, which we all agree there is a tremendous amount of promise behind the cell therapies, and we see a great deal of commercialization in the near future around several cell and gene therapies, and understand that this is going to place a great deal of strain on the supply chain. But also on the downsides, right? We have to be able to build a system that is impervious to different disruptions, to different sources of fragmentation. And COVID-19, I think, is the nearest example that we all have at hand and are currently living under. And we need to find a way to learn from the experience that we're all living under to create a more robust supply chain at the end of the day. Now, I think that we consider the issues around resilience to be important, both in terms of standardization, in terms of the way we design these supply chains, and in terms of how we build regulatory compliance around these supply chains. Next slide, please. Ultimately, it's on all of us to make sure that the resilience that we build into our supply chains is robust, that we make sure that we address all of the both upside and downside associated with the challenges we're going to face, and really build it from the ground up together. So it's going to come down to standardization. It's going to come down to creating interfaces between us and the industry, suppliers and cell therapy developers, advanced therapy developers, that is able to really stand up as more of these go commercial. It's going to mean building these supply chains from the very beginning in a way that allows for redundancy and interoperability. And it's going to mean building highly compliant ancillary materials that are important for us to commercialize these therapies on time and in a way that is supportive of effectiveness and safety for patients. So thank you so much for your time. Very much appreciated. I look forward to any questions you might have as we move along.
Thanks a lot, Ezequiel. A very interesting presentation. I don't see questions coming from the audience, so I have a couple of questions for you. And you mentioned in your presentation the challenges and disruptions, and making as an example the COVID-19 crisis. Is there, from your perspective, anything that you can add in that respect, and how the COVID crisis has put constraints or on the manufacturing industry and on your side of course of defense?
Yeah, no, that's a great question, Paulo. I think it's something that we're all facing. I think when you look at key consumables, bags, I think is a great example. bioprocessing bags are really being driven in large part towards the production of vaccines and COVID-19 relevant therapies. And so what that does ultimately is build out lead times for everyone else. And so we are at the same time working very closely with our suppliers, with our secondary suppliers, to make sure that we experience as little disruption as possible, considering the constraints. But I think the clearest example that I can think of, and that folks in the audience are probably well aware of, is constraints around primary packaging, around caps, around vials, around bags, because these are all being driven elsewhere. And I think we just need to find a way to adjust to make sure that we have secondary suppliers onboarded, that we're building up sufficient stock to be able to deal with these disruptions, because otherwise we are going to be caught flat-footed.
Thanks. That's very helpful. And do you think, let's say, in terms of manufacturing capacity and all the related technologies and supplies, there will be, do you see challenges from the growth of the cell and gene therapy industry, or cell therapy in your case? How do you see that feasible in the years to come? Let's say, so both from the crisis and the growth of the sector. Is the trajectory is feasible in your view?
Yeah, I think it is. But I think, and it comes back to something I talked about earlier on in terms of standardization. If we don't achieve the degree of standardization we need, it's going to be very, very difficult to scale these therapies. At the end of the day, if you look at any other industry, whether it's the automotive industry, the semiconductor industry, agricultural industry for that matter, there's a great deal of work that happens in the background to standardize and to make sure that the interfaces between different suppliers and different manufacturers is clear. Because if those interfaces are not clear, well-defined, and understood, and I think that the right place for this is in standard-setting organizations that operate in the US, in Europe, globally. If those standards aren't built from the ground up, and if we don't build an industry that understands what they are and build them into their processes, we are destined to remain a bespoke industry that is unable to really deal with the demand that we're likely to face for these life-saving treatments. These treatments really promise to change the way that we treat people and cure people. And I think that if we're unable to achieve a supply base and a manufacturing platform that is truly scalable, we're unlikely to be able to realize that promise.
Thanks, very helpful. And just another question that comes out of what you're saying. Let's say you're mentioning standardization and also scale. So it makes me think. So the next question is about, how do you see these trends impacting COGS? Let's say, are these volumes going to have an impact on manufacturing costs overall and specifically on supplies?
Yeah, and I think that's ultimately where everyone's driving towards, right? Because at the end of the day, beyond kind of supply chain integrity, it's going to be COGS that is going to drive uptake on a lot of these, especially in certain geographies where you're going to have payers that have a great deal more purchasing power and ability to drive down purchasing prices. So I think that we as an industry right now haven't focused exclusively on COGS because we're trying to build therapies from the ground up. But as we move forward, it's going to be important that we consider this more and more. So again, from a cytokine production perspective, we're able to scale our production capacity to a place where we're able to support several leading clinical and commercial immunotherapy companies, understanding that the requirements are for large-scale, consistent production. And we think that that's going to be more and more often the case. Now, what that allows us to do is to really drive down cost of production as we continue to scale and ensure that we're able to support the commercialization of these therapies at a COGS that makes sense. But ultimately, it's going to be on the cost of ancillary materials. It's going to be on the robustness, automation, and closure of the manufacturing process. And an item that we haven't talked about so much, but really the shift in modalities from an autologous perspective into an allogeneic and iPSC derived. So I think that there are shifts in the industry that are going to continue to drive down COGS, and we need to be mindful that these changes are not going to be linear, but that they all will drive down the cost of producing these therapies. And we all need to be a part of that, because ultimately we are going to be as successful as we can make these therapies cost effective.
Thanks a lot, Ezequiel. Really very nice presentation and chat with you. We are at the end of our half an hour together. So I'd like to thank you again for this. Hope to see you soon at next conference or in person eventually.
Thanks, Paolo. Much appreciated. Thank you, everyone.